Hello. First, I would like to thank the organizers for giving me the opportunity to present our work today. And My name is Samer Hussein, and I'm an associate professor at Université Laval. Today, I'll talk to you about a long-encoding RNA that we've been working with for the past couple of years, and we think it's really implicated in regulation of pluripotent stem cell states. Our our work in the lab focuses on the different functions of or mechanisms of long-encoding RNAs, more specifically using two models. One is looking using pluripotent stem cells and see how long-encoding RNAs affect development and cell state changes, but also cancer models like glioblastoma or brain tumors to see how long-encoding RNAs affect disease progression. The long-encoding RNAs, as you might all know, are classified as RNAs of 200 nucleotides or more, and they're often capped. They're polyadenylated. They're transcribed by RNA polymerase II, spliced, and generally they're about 10 times less expressed than mRNAs, and they are also more tissue specific than mRNAs. They have been implicated in several functions in the cells. In the nucleus, they can regulate chromatin state and gene expression through the recruitment of chromatin modifying complexes and transcription factors and so on. They have been shown to be implicated in nuclear structures such as nuclear bodies and RNA splicing. But also in the cytoplasm, they have been shown to affect mRNA translation and mRNA stability. In the lab, we have three projects that touch on different aspects of long-encoding RNAs. But today, I would like to focus on one of my PhD students' projects, Victoire Faure. And this is a long-encoding RNA that we think may be implicated in alternative splicing in pluripotent cells. We use the embryonic stem cells as a model. Embryonic stem cells basically can be grown for long extended periods of time and can differentiate towards all three germ layers and essentially any cell type of the body to form the embryo and also the whole organism. What is nice is now we can actually take any differentiated cell, reprogram it back to what we call a pluripotent stem cell or induced pluripotent stem cell, iPSCs, using only a cocktail of 4 transcription factors, OCT4, KLF4, SOX2, and c-MYC. And I have spent quite a bit of my postdoctoral years really studying this process. And from data that we have accumulated on reprogramming, I was able to identify one long-encoding RNA that could be implicated in reprogramming and ES cells. What we call Tapir, and I'll go towards the end, I'll tell you why we call it Tapir. And this long-encoding RNA is specifically expressed in ES cells and iPS cells and is also nuclear. There's a couple ways to study the function of a long-encoding RNA in pluripotency. One being through the reprogramming, where you overexpress your long-encoding RNA of interest in differentiated cells to see if you can have more iPS cells or more pluripotent stem cells emerging when you overexpress it, suggesting that it's like a gain of function technique. Or you can knock it out or knock it down in ES cells to see if it causes the differentiation of these cells. Today I'll focus mostly on knockdown and knockout of Tapir in ES cells. So for knockdown techniques, we use the CRISPR-I system, which is a DETCAS9 fused to a CRAB inhibition domain. And basically with a guide that targets your sequence of interest, here we're targeting the transcriptional start site of Tapir, you inhibit the transcription of Tapir. And we have an inducible system where we induce the expression of the CRISPR-I. So we introduce these plasmids and the guides, Tapir targeting guides and non-targeting guides as control in ES cells. And we select to have the cells that are expressing, that are containing these plasmids, and we induce with docs the expression of the CRISPR-I, and we extract RNA at day 0 and at day 3 and at day 5 and see really how it affects the cells. What we found using long-read RNA sequencing of the Tapir knockdown ESCs, we found that by day 5 of Tapir knockdown, we see that the cells have shifted from a naive pluripotent state, which is the ES cell state, to a primed pluripotent state, which is an APLC-like cells, suggesting that Tapir knockdown causes the differentiation of the cells. And when we look globally at the RNA sequencing data for biological processes, we noticed that several developmental processes or genes that are implicated in developmental processes are affected or upregulated when we knock down Tapir, suggesting that it promotes differentiation of the cells again. More so, also, you can see that the Wnt pathway, genes implicated in the Wnt pathway are also in this case, down-regulated, suggesting that emphasizing how Tapir is promoting this naive to primed pluripotency transition. One interesting thing that came out of the data also is the keyword, the uniprot keyword, the alternative splicing, and I'll get to that later in my talk, how Tapir might be implicated in alternative splicing. So we all know that when we use any knockdown technique, you never knock down the gene 100%. So we wanted to see if we can actually do it, completely knock out the Tapir function. So what we did, we targeted exon 4, which covers most of the Tapir sequence, and with Cas9 to completely remove it. And while doing the removal of exon 4, we also introduced a control plasmid expressing an antisense of the luciferase gene, so it's just a random RNA, and also the transgene, the gene Tapir, the sequence of Tapir to express the C Tapir at the same time. Because essentially sometimes in ES cells, you cannot knock out the gene because it affects pluripotency. So in this case here, by maintaining Tapir expression and then removing DOX, we completely remove the expression of Tapir just by removing DOX. So we can see we selected several clones, or Victoire selected several clones, and by analyzing a few of them here, you can see that the wild type band is around 1900 base pairs, when we delete the allele completely, you have about 430 base pairs. What was really interesting is we could not get a knockout with the control RNA, but only when Tapir was expressed the Tapir transgene was expressed, that we can actually obtain knockout, suggesting that Tapir is required for the maintenance of ES cells. So in summary, basically, when we knock down Tapir, we cause differentiation or loss of ES cells. One thing I haven't talked about, and this is in my poster, if you'd like to come later, we can discuss it more. Essentially, if we add Tapir during the reprogramming process, we can see that we induce quite a bit of formation of iPS cells, actually drastic improvement in the reprogramming process when we add Tapir. Suggesting Tapir is really implicated in pluripotency and more so in the acquisition of pluripotency. Now we wanted to study the function of Tapir. And essentially one way to study a function of long-encoding RNA is to use this technique, the chromatin isolation by RNA precipitation, CHIRP, to see what are the interactors with our long-encoding RNA and where they interact on the genome, if they do interact on the genome. So we can isolate by pulling down with Tapir probes and using as a control like Z probes. We can check the genomic DNA from the pull downs, proteins that are complexing with the RNA or the whole complex of the RNA, DNA, and our proteins, as well as RNA. So today it's mainly on RNA interactions with Tapir. So when we did the Tapir and looked at the RNAs that were pulled down with it, we found that many genes that are like HMT2, KDM5A, DNMT1, many genes that are implicated in chromatin binding were being pulled down with Tapir, but also many pluripotency associated genes like ESRB, NMYC, REX1, KLF5, and DPPA5. Those are all well-known genes that are important for regulating the pluripotent stem cell state. And some of the genes that were implicated in cadherin binding and cell-cell adhesion, which is an important process for maintaining ES cells, were also pulled down with Tapir. So it seems to have a big effect on or interacting with many genes that are implicated in pluripotency. When we look at the expression of these genes in our knockdown, we don't see a difference. So this was a bit surprising to us. So it made us think maybe, okay, we should look at the RNAs if there's something happening at the RNA level rather than the expression of the RNA. And when we look more specifically at certain genes that we've pulled down, we noticed that for example, here REX1, which was also identified in RNA interactome genome-wide with a technique called Mario, when we knock down Tapir, we see intron retention within the first intron of REX1. We're also using lncRNA, long read RNA sequencing. We can actually identify new novel exons that we couldn't identify before. We notice also alternative 3' UTR length differences. For example, here's MAPK1, and when we knock down Tapir, you have a different 3' UTR length. You have half the transcripts are a shorter 3' UTR. Also, several of the genes that were pulled down, like NMYC and EHMT2, were also affected, when we knocked down Tapir, having more different exons or a new exon being alternatively spliced, or exon skipping as well. So we decided, okay, we're going to look at the sequence of Tapir and see what sequence could be giving the same, if we find similar sequences between the RNAs that are being pulled down and Tapir. And we found one Sine B2 element within the Tapir exon 4, which is basically Sine elements cover about 10% of the genome, and they around 80% of long-encoding RNAs actually have retrotransposable elements within them. So this was interesting. When we look at the RNAs that were pulled down with Tapir, we can see that there's many inverted Sine B2 elements within the genes that were pulled down, like here in the 3' UTR and introns, and also ones that were detected by Mario. The binding of Tapir was within this inverted Sine element, suggesting that it's through complementary sequences that Tapir is pulling down these, or is binding to these RNAs and affecting their function. Another interesting aspect that we found during the Tapir knockdown is there was an overall higher expression of Sine elements by themselves, which is odd because usually they should not be expressed, but when we knock down Tapir, we see these elements being expressed. When we look globally at intragenic repeat elements and intergenic, we can see that the differentially expressed repeat elements are mostly upregulated upon Tapir knockdown, specifically the B2MM1A, which is the Sine B2 element within Tapir, is really upregulated in whether it's intragenic or intergenic. So that made us, okay, to really address the function of the Sine element in ESL. So we went back to our knockout model where we overexpress our transgenes while we knock out the exon 4 of Tapir. And when we co-transfect with Tapir, when we co-transfect with the Cas9 and the guides, plus our transgene for a Tapir transgene that is devoid of the Sine element, so we transfect these in ES. What we find is, again, we cannot obtain a knockout, suggesting that the Sine element within Tapir is really responsible for the function of Tapir, and really this Sine element is the functional aspect of functional portion of the Tapir sequence that we have. So we did also experiments with reprogramming, and if you reprogram towards iPSCs with this construct that is devoid of the Sine element, we can see that we abolish this increasing of reprogramming that we see with Tapir wild type sequence, suggesting again that the Sine element is important for the function of Tapir. So our working model is that Tapir, through the inverted elements within its mRNA targets, binds through the Sine element, and we think that it brings in mRNA processing regulators that are maybe implicated in splicing and so on, that affect prepotency genes and chromatin remodeling mRNAs, and this is how Tapir controls prepotency. And one of the reasons why we call it Tapir is really because Tapir is a favorite animal of Victoire, and also because it fits with the name that we gave it, which is transposon associated prepotency inducing RNA. With this, I'd like to thank first of all the people that did the work, Victoire Faure, and Gabriel Khalifi. And the awesome team that I have, really, they work hard, and we've been working hard on different parts of the long coding RNAs, and also the funding agencies with their support, which the work couldn't have been done without their support. And I want to thank also everybody that's listening to this presentation, and the organizers again for giving me this opportunity. Thank you, and have a good day.